Okay, let's unpack this. Welcome to the Deep Dive, the show where we tackle complex topics, arm ourselves with the best research, and cut through the noise to get to the core of what you need to know. Today, we're plunging into a subject that touches nearly everyone, directly or indirectly: cancer. It's a word that evokes, well, a lot of complexity, but our scientific understanding has actually grown dramatically. Our mission for this deep dive is to explore the biology of cancer, drawing exclusively from the chapter on cancer biology in Understanding Pathophysiology, 7th edition. This is a focused journey, custom tailored to help you, maybe you're a college student grappling with these concepts, grasp at the core mechanisms and clinical aspects without getting, overwhelmed.
What's truly fascinating here is how decades of research have really reshaped our understanding. The key insight, I think, is that cancer isn't one enemy, but a vast army of over 100 different diseases. Each one has its own unique genetic and epigenetic fingerprint, which means our fight against cancer has to be as personalized as, well, our own biology. It's this complex interplay of environment, heredity, and even our own behavior. Our goal today is to provide a clear, step-by-step understanding of this topic, guiding you through the chapter's most important insights.
That's a powerful start. So let's begin right at the beginning. How do we actually define cancer today? And where does that powerful word even come from historically?
Well, the National Cancer Institute defines cancer as diseases in which abnormal cells divide without control and are able to invade other tissues. Pretty straightforward definition, right? The term cancer itself has a rich history. It comes from the Latin translation of the Greek carcinoma. Hippocrates used it to describe these, like appendage-like projections extending from tumors, reminded him of a crab. And interestingly, the word tumor originally just meant any swelling, but today we generally reserve it for a new growth, or neoplasm. And this brings us to a really crucial distinction: benign versus malignant.
Ah, yes, that distinction is absolutely foundational. So let's break down the differences, starting with what we consider benign.
Okay, so benign tumors are generally, let's say, well-behaved. Imagine them like a perfectly round bubble. They're usually encapsulated by connective tissue, giving them a clear, defined boundary. You can sort of see where they start and end. The cells inside are well differentiated, meaning they still look a lot like the normal tissue they came from, and their internal structure, the stroma, is pretty well organized. Crucially, they don't invade beyond that capsule, and they don't spread to distant locations. No metastasis. Cell division is very rare. We often name them with an oma suffix. So a leiomyoma is a benign tumor of smooth muscle. A lymphoma comes from fat cells. Now, while benign sounds harmless, it's important to remember they can still be dangerous depending on location. A meningioma, a benign brain tumor, can grow large enough to compress vital brain tissue. That could definitely be life-threatening.
Right, so benign means contained, but not necessarily benign in impact. Okay, what about the truly aggressive forms, the malignant tumors?
Yeah, malignant tumors, the cancerous ones, are a whole different story. They typically have much more rapid growth rates. Microscopically, they show significant alterations like anaplasia, which is basically the loss of cellular differentiation. The cells lose their specialized features, look more primitive, kind of disorganized. They're also pleomorphic, meaning they show marked variability in size and shape. So unlike orderly rows of normal cells, malignant cells are haphazard, irregular, wildly varied. Their nuclei are often large and darkly stained, and you see active cell division much more commonly. They lack that capsule, there's no protective boundary, and they aggressively invade nearby blood vessels, lymphatics, and surrounding structures. But their most dangerous characteristic, really, is their ability to spread far beyond their tissue of origin, that's metastasis.
That invasive spread is truly terrifying. How do we even begin to classify these aggressive diseases then?
Cancers are generally named according to the cell type they originated from. So if they arise from epithelial tissue like skin or linings of organs, they're called carcinomas. For example, a mammary adenocarcinoma comes from breast glandular tissue. Cancers from mesenchymal tissue, that's connective tissue, muscle, bone, are usually sarcomas, like a rhabdomyosarcoma in muscle. Cancers of lymphatic tissue are lymphomas, and those of blood-forming cells are leukemias.
Okay, got it. And what about those tricky precancers we sometimes hear about, like things that aren't quite cancer yet?
You're talking about carcinoma in situ, or CIS. These are essentially preinvasive epithelial tumors. They're localized to the epithelium, meaning they haven't penetrated the basement membrane or invaded the surrounding stroma yet. They're not technically malignant, but they're absolutely critical to detect because they represent a very early stage of cancer development. If you picture it, imagine abnormal cells sitting right on top of that smooth, protective basement membrane, but they just haven't broken through that barrier. CIS lesions can do one of three things. They can stay stable, they can progress to invasive cancer, or sometimes they even regress. The PAP test, for instance, is incredibly effective because it catches cervical CIS early, often years before it might progress.
So these classifications aren't just academic jargon. They're really the first crucial steps in understanding how to approach a specific cancer, guiding everything from diagnosis to treatment, right?
Absolutely. It sets the stage for everything that follows.
Okay, here's where it gets really interesting, I think. Now that we have the basic language down, let's dive into the core biology. You mentioned Hanahan and Weinberg earlier, and their hallmarks of cancer. Can you unpack that a bit? It sounds like a kind of instruction manual for how a cell becomes cancerous.
That's a great way to put it. Yeah, their work, starting with 6 and now expanded, really provides a framework for understanding the fundamental changes a cell undergoes to become malignant. The big picture is that cancer is incredibly complex, and it thrives by constantly adapting its strategies within this dynamic environment, the tumor microenvironment we touched on. You can kind of think of cancer development in three broad stages. Tumor initiation, that's the initial genetic or epigenetic hit. Then tumor promotion, where that altered cell population expands. And finally, tumor progression, leading to invasion and spread, metastasis. And critically, cancer incidence increases dramatically with age, largely because we accumulate these genetic changes over a lifetime.
And these genetic changes are really the root of how cancer gets started, aren't they? What exactly are we talking about when we say genetic changes? Is it just typos in the DNA?
Well, it's more than just typos, though. Point mutations changing a single DNA base pair, like that single letter change in a book, are definitely part of it. But we also see larger scale changes, chromosomal translocations, where pieces of chromosomes swap places. Think cutting and pasting a whole paragraph to a different page. And then there's gene amplification, where a section of a chromosome gets copied over and over again, like photocopying one paragraph hundreds of times, making way too much of whatever that gene codes for. But it's not just changes to the DNA sequence. We also have epigenetic effects. These are things like DNA methylation or histone modifications that change how genes are read, turning them on or off, without actually altering the underlying DNA code itself. It's like adding sticky notes to the book that change how you read it.
So not all mutations are created equal then. Some are driving the bus, others are just along for the ride?
Exactly. We talk about driver mutations, which actively push the cancer progression forward, versus passenger mutations, which are sort of random background noise that don't really contribute. Once a cell accumulates a critical number of these driver mutations, it gains a selective advantage. Its descendants, its progeny, multiply faster than their neighbors. This is called clonal proliferation or expansion. Picture a diverse crowd of cells, maybe in your colon lining. Then imagine one specific cell gets a mutation, maybe in the APC gene, and it starts dividing a bit faster. Then one of its descendants gets another hit, maybe in RAS, and starts dividing even faster, forming a small polyp. It keeps accumulating hits. P53 loss is often a late one, and eventually one lineage dominates, becoming invasive cancer.
Wow. So it's this stepwise accumulation, and it's not just about the cancer cells themselves, but also how they interact with everything around them, that tumor microenvironment you mentioned.
Precisely. The whole process is, in many ways, like a wound that just never heals. The growing cancer cells and even nearby normal cells they influence start pumping out pro-inflammatory signals. These signals recruit all sorts of cells normally involved in tissue repair inflammatory cells like macrophages, immune cells, fibroblasts. These recruited cells form the stroma, the supportive tissue, or the tumor microenvironment. And this stroma can actually make up a huge chunk of the tumor mass, sometimes up to 90%. There's constant back and forth communication, extensive signaling, and the cancer cells get really good at manipulating these normal stromal cells to help them grow, invade, and hide from the immune system. So yeah, think of the tumor less as a solid block of bad cells and more like a bustling, corrupt ecosystem where the cancer cells are constantly manipulating their neighbors for their own gain. Okay, so we've got this picture of cancer as this constantly evolving, genetically unstable entity manipulating its surroundings. Now let's zoom in on those specific survival strategies, the hallmarks themselves. Where do we start? Let's start with the most fundamental one. Sustained proliferative signaling, basically uncontrolled growth. Normal cells only divide when they get specific signals, growth factors. Cancer cells figure out ways to keep that growth signal permanently switched on. The normal genes that regulate proliferation are called proto-oncogenes. When they get mutated or overexpressed, they become oncogenes, and that's like having the gas pedal stuck to the floor. How do they do it? Well, several ways. Some cancer cells start making their own growth factors. That's autocrine stimulation. Others develop way too many growth factor receptors on their surface, making them hyper-responsive. The classic example is HER2 amplification in some breast cancers. Even a single point mutation like in the RAS gene, common in pancreatic and colorectal cancers, can jam the signaling pathway in the on position. Or you can get those big chromosomal translocations. The famous Philadelphia chromosome in chronic myeloid CML creates a brand new fusion protein, BCR-ABL, which is a constantly active enzyme driving cell division. That discovery, by the way, led directly to targeted drugs like imatinib. They can also use gene amplification, just making hundreds of copies of an oncogene like NMYC in neuroblastoma, massively boosting the growth signal.
Okay, so oncogenes are like flooring the gas pedal. But what about the brakes? Because normally our cells have powerful mechanisms to stop runaway growth, right?
Absolutely right. That's the next hallmark, evading growth suppressors. Our cells have tumor-suppressor genes, sometimes called anti-oncogenes. These are the brakes. They normally regulate the cell cycle, inhibit proliferation, stop division if there's DNA damage, and prevent mutations from accumulating. For cancer to really take hold, these brake systems usually need to be inactivated. And critically, because we inherit two copies of most genes, one from each parent, you generally need two hits, two mutations, one in each allele, to completely knock out a tumor suppressor gene's function. This is different from oncogenes, where often one hit is enough. A key example is the tumor protein, P53, TP53 gene. It's often called the guardian of the genome. What P53 does is monitor the cell for stress, things like DNA damage or lack of oxygen. If it detects problems, it activates caretaker genes, the cell's DNA repair crew. If the damage is too severe to fix, P53 can trigger apoptosis, program cell death, basically telling the cell to self-destruct for the greater good. But if you lose P53 function, damaged cells don't get repaired or eliminated. They just keep dividing and accumulating more mutations, which massively increases cancer risk. Think of P53 as the cell's internal quality control inspector. Without it, errors just pile up unchecked. This two-hit idea also explains why inherited mutations in one copy of a tumor suppressor gene, like RB1 for retinoblastoma, or P53 in Life-Romany syndrome, or BRCA12 for breast ovarian cancer risk, dramatically increase susceptibility. You're already born with one brake malfunctioning, so you only need one more somatic hit to lose the function entirely.
Wow. Okay, so that raises a crucial question. If mutations are happening constantly and the brakes are potentially failing, how does the cell normally handle all this? And what happens when it, well, basically becomes immortal?
Right, that leads us straight into two interconnected hallmarks: genomic instability and enabling replication immortality. Genomic instability just means there's an increased tendency for mutations and alterations in the genome to occur and persist. This often happens because those caretaker genes, the DNA repair crew we just mentioned, get mutated themselves. TP53 plays a role here too, but also genes like BRCA1 and BRCA2, which are crucial for repairing complex DNA breaks. If you inherit mutations in these caretaker genes, like in Lynch syndrome related to mismatch repair defects, or in BRCA carriers, your cells are just much worse at fixing DNA errors, leading to instability and higher cancer risk. Cancer cells also use epigenetic silencing to turn off tumor suppressors, and they can hijack small RNA molecules called microRNAs. Some of these, called oncomeres, actually promote cancer when they're dysregulated. Then you have replication immortality. Normal cells aren't designed to live forever. They have a built-in clock, often called the Hayflick limit. They can only divide a certain number of times, maybe 50 or 60, before they stop or undergo apoptosis. Part of this clock involves the shortening of telomeres. Telomeres are protective caps at the ends of our chromosomes. Think of them like the plastic tips on shoelaces. They prevent the ends from fraying or sticking together inappropriately. Every time a normal cell divides, these telomeres get a little bit shorter. Eventually, they get critically short, signaling the cell to stop dividing. Now, there's an enzyme called telomerase that can rebuild these telomeres. It's usually active in germ cells and some stem cells, but mostly turned off in our regular body cells. But what do cancer cells do? About 90% of them find a way to switch telomerase back on. This rebuilds their telomeres, effectively making them immortal. They bypass the Hayflick limit and can just keep dividing indefinitely. It's like they have a perpetual shoelace tip replacer.
Okay, so cancer cells are immortal, unstable, constantly pushing for growth. But surely they need resources, like fuel and oxygen to keep that engine running so hard. How do they manage that?
Absolutely, they become very demanding. And that brings us to the next hallmark, inducing angiogenesis. Angiogenesis is just the formation of new blood vessels. See, a tumor can only grow to about 1 to 2 millimeters in diameter, like the head of a pin, before it outstrips its oxygen and nutrient supply just by diffusion. To get any bigger, it absolutely needs its own dedicated blood supply. So cancer cells start secreting signals, angiogenic factors like VEGF, vascular endothelial growth factor, and BFGF, that encourage nearby blood vessels to sprout new branches toward the tumor. They also suppress natural inhibitors of angiogenesis. Low oxygen levels, hypoxia, within the growing tumor actually trigger a factor called HIF-1, which ramps up the production of these pro-angiogenic factors even more. And importantly, the blood vessels that grow in response to these tumor signals are often abnormal. They're leaky, disorganized, tortuous, and prone to hemorrhage. But this leakiness also provides an easy route for cancer cells to escape into the circulation. Think of a normal, orderly tree branching out, and then compare it to this chaotic, leaky tangle of pipes forming haphazardly around the tumor.
Right, building their own supply lines. And this leads us to another really fascinating adaptation, how they actually fuel all this crazy growth.
Yeah, the metabolism of cancer cells is quite different. This is the hallmark of reprogramming energy metabolism. Cancer cells need a lot of building blocks, lipids, nucleotides, amino acids, to make new cells rapidly. Way back in the 1920s, Otto Warburg noticed something odd. Even when there's plenty of oxygen around, many cancer cells seem to prefer a less efficient way of making energy called glycolysis, rather than the usual, more efficient oxidative phosphorylation that happens in the mitochondria. This is known as the Warburg effect, or aerobic glycolysis. Seems counterintuitive, right? Why choose a less efficient path? Well, the key insight is that while glycolysis produces less ATP per glucose molecule, it does so very quickly, and crucially, it shunts metabolic intermediates into pathways that generate the building blocks needed for rapid cell proliferation. So it's less about maximizing energy yield and more about maximizing biomass production. Clinically, this high glucose uptake is so characteristic that we exploit it for diagnosis. PD scans using 18F-fluorodeoxyglucose, FDG, work because cancer cells gobble up this glucose analog much faster than most normal tissues. Those bright glowing spots you see on a PET scan, that's often cancer, furiously consuming glucose like tiny greedy engines.
Wow. So they're feeding themselves, building their own blood supply. Okay, but what about our body's own defenses? Surely our immune system is designed to spot and destroy these rogue cells.
It is, absolutely. But cancer cells become masters of resistance to destruction. This is a big one, encompassing several key strategies. First, they learn to resist apoptotic cell death. Apoptosis, that programmed self-destruction we talked about with P53, is a fundamental defense. Cancer cells find ways to disable this pathway, often by losing P53 or by overexpressing anti-apoptotic proteins, basically refusing to die when they should. Second, they actually corrupt inflammation to their advantage, tumor-promoting inflammation. We know chronic inflammation is a risk factor for many cancers. Think of Helicobacter pylori infection causing chronic gastritis, which increases risk for gastric cancer. Tumors actively manipulate the inflammatory response. Instead of an anti-tumor attack, they shift it towards a pro-wound healing tissue regeneration phenotype, which actually promotes their proliferation, angiogenesis, and even suppresses the immune attack. Tumor-associated macrophages, TAMs, are key players here. They get recruited to the tumor, but instead of attacking, they often block other immune cells and secrete growth factors that help the tumor. Third, and this is huge in modern therapy, they figure out how to evade immune destruction. Our immune system, particularly T cells and NK cells, should recognize cancer cells because they often express abnormal proteins, tumor-associated antigens. The whole idea of immune surveillance is that our immune system usually mops up nascent cancers. And we know this works sometimes. Vaccines against cancer-causing viruses like HPV for cervical cancer and HPV for liver cancer are incredibly effective cancer prevention tools because they let the immune system eliminate the threat early. But established cancers develop clever evasion tactics. They can produce immunosuppressive factors like TGF-beta. They can recruit inhibitory immune cells like T regulatory cells, TREGs, that actively shut down the anti-tumor response. They can stop expressing the antigens the immune system recognizes or hide the molecules needed to present those antigens. It's like the cancer cell is wearing different disguises or putting up a shield to deflect immune attacks.
Okay, so if a cancer cell has managed all that evaded the brakes, become immortal, built its own roads, found fuel, and learned how to hide from the cops, the immune system, what's its ultimate, most dangerous move?
That would be the final hallmark. Activating invasion and metastasis. Invasion is local spread, breaking out of the original tissue. Metastasis is the spread to distant sites through the bloodstream or lymphatic system. And metastasis is unfortunately the major cause of death from cancer. It's actually a remarkably inefficient process millions of cells might shed from a primary tumor, but only a tiny fraction will successfully establish a metastasis. But the ones that succeed are obviously extremely dangerous. To do this, cells first have to invade locally. This involves breaking loose from their neighbors, often by downregulating adhesion molecules like E-cadherin. They then have to digest the surrounding tissue matrix using enzymes like matrix metalloproteinases, MMPs. They need to become motile. A key process here is called epithelial mesenchymal transition, EMT. Epithelial cells, which are normally stuck together and stationary, undergo changes to become more like mesenchymal cells, migratory, invasive, and resistant to death. Then they need to get into a blood vessel or lymphatic vessel intravasation. The leaky neovessels they induced earlier provide easy access. They have to survive the journey in the circulation, which is hostile territory. They often cloak themselves by sticking to platelets. Then they have to get out of the vessel at a distant site, extravasation, and survive in a new environment. Metastasis isn't random. Cancers often show preference for specific organs. This is called organotropism. For example, colon cancer often goes to the liver, breast cancer to bone, lung, liver, brain. This likely involves specific molecular interactions between the cancer cells and the target organ. And finally, sometimes cells arrive at a distant site but remain dormant for years, not growing into a detectable metastasis cellular dormancy. This is a huge clinical challenge because dormant cells are often resistant to current therapies. So yeah, metastasis is like this incredibly complex multi-step obstacle course or military operation, breaking ranks, tunneling through defenses, traveling through hostile territory, surviving, and then, if conditions are just right, setting up a new outpost far away.
Wow, that whole picture of the biology is just incredibly complex. But understanding it helps make sense of the symptoms people experience and ultimately how we try to fight back. So, beyond the microscope, how does cancer actually show up in a person?
Right, the clinical manifestations are diverse. One really interesting category is paraneoplastic syndromes. These are symptom complexes that are triggered by the cancer, but aren't caused by the tumor physically being there or invading tissue. Instead, they're caused by substances the tumor releases, maybe hormones, cytokines, or sometimes by an immune response to the tumor that cross-reacts with normal tissues. For example, some small cell lung cancers secrete ACTH, a hormone, which leads to Cushing's syndrome. Other cancers might cause hypercalcemia or neurological problems. These syndromes can actually be a very first sign of an unknown cancer, and they can be quite serious in their own right. Then, of course, there are the more common side effects, both from the cancer itself and from its treatment. These are things you will definitely encounter. Anemia is very common due to chronic bleeding, malnutrition, the cancer-affecting bone marrow, or chemotherapy. Cachexia is particularly devastating. It's a severe wasting syndrome, profound loss of muscle and body fat, inflammation, loss of appetite. It's not just simple starvation. The tumor actively hijacks the body's metabolism. It could be irreversible and is a major contributor to mortality. Fatigue is probably the single most frequently reported symptom, just profound, debilitating tiredness. The gastrointestinal tract often takes a hit because its cells divide rapidly, making them vulnerable to chemo and radiation. This leads to things like mouth sores, diarrhea, malabsorption, and increased infection risk. Hair loss, alopecia, is a well-known side effect of many chemotherapies, though usually temporary. Radiation causes hair loss only in the treated area. Infection is a huge risk and a leading cause of complications and death, mainly because both the cancer and its treatments can severely suppress the immune system. Leukopenia, low white blood cells, and thrombocytopenia, low platelets, are common results of chemo or radiation hitting the bone marrow, increasing risk of infection and bleeding, respectively. And finally, pain. While not always present early on, pain often develops in later stages due to the tumor pressing on nerves, obstructing organs, invading tissues, or causing inflammation.
Okay, so those manifestations are often what bring patients into the clinic. How do clinicians then actually diagnose the cancer and figure out how advanced it is and then, decide how to treat this incredibly cunning disease?
Diagnosis fundamentally relies on getting a piece of the tissue, a biopsy, and having a pathologist examine it under a microscope. That's the gold standard. There are different ways to get that tissue: surgical excision, needle biopsies, etc. And there's growing excitement about liquid biopsy. This involves looking for circulating tumor cells or fragments of tumor DNA or RNA in a simple blood sample. The hope is it could allow for earlier detection, monitoring treatment response, and detecting recurrence less invasively. Still evolving, but very promising. Once cancer is confirmed, staging is absolutely critical. Staging tells us how far the cancer has spread, and that heavily influences treatment decisions and prognosis. We generally use a 4-stage system, where stage 1 is localized and stage 4 means distant metastasis. Carcinoma in situ is often considered stage 0. The most widely used system is the T&M system. T describes the size and extent of the primary tumor and indicates whether the cancer has spread to nearby lymph nodes, and M tells us if there's distant metastasis. Higher T, N, and M stages generally mean a worse prognosis. So a doctor might describe a breast cancer as T2N1M0, meaning a certain size tumor, spread to nearby nodes, but no distant spread yet. We also use tumor markers. These are substances, hormones, enzymes, antigens produced by the tumor or by the body in response to the tumor, which can sometimes be detected in blood, urine, or tissue. They can be useful for screening certain high-risk groups, helping diagnose specific cancer types, or, very importantly, tracking how well treatment is working or detecting recurrence. But, and this is a big caveat, no single tumor marker is perfect for screening the general population. There are issues with false positives, false negatives, and non-cancerous conditions causing elevated levels. The debate around PSA screening for prostate cancer highlights these complexities. Increasingly, though, diagnosis is moving towards personalized medicine. We're analyzing the tumor tissue not just microscopically, but also for specific protein expression, like hormone receptors or HER2 in breast cancer, and genetic mutations. This allows for much more precise classification and helps tailor treatment directly to the tumor's specific biology.
Right. That makes perfect sense. So once we know what kind of cancer it is, how far it's spread, and maybe even its specific molecular fingerprint, what are the main ways we actually try to fight it?
Well, the classic approaches, the cornerstones for many years, have been surgery, radiation therapy, and chemotherapy. Surgery is often the main treatment for solid tumors that haven't spread, aiming for complete removal. It can also be palliative, to relieve symptoms, or prophylactic, preventive, like removing the colon in someone with a high-risk genetic syndrome. Radiation therapy uses high-energy rays to damage cancer cell DNA and kill them. It's particularly good for localized disease. It could be delivered from outside the body, external beam, or by placing radioactive sources directly inside or near the tumor, brachytherapy. Chemotherapy uses drugs that target rapidly dividing cells. Since cancer cells typically divide faster than most normal cells, they're more susceptible. The field actually has roots in observations made with mustard gas during wartime. Chemo drugs work in different ways. Some interfere with DNA replication, some damage DNA directly, some mess with the cell's structural components. A key principle is combination therapy, using multiple drugs with different mechanisms to hit the cancer from different angles, hopefully increasing effectiveness and reducing the chance of resistance. Chemo can be used to shrink a tumor before surgery, neoadjuvant, kill any microscopic cells left after surgery, adjuvant, or as the main treatment to induce remission, induction. The major downside, of course, is that chemo also harms normal, rapidly dividing cells in the bone marrow, hair follicles, gut lining, causing those well-known side effects. But a landscape is rapidly changing with modern and evolving approaches. Immunotherapy is arguably the most exciting area right now. The goal is to harness the patient's own immune system to fight the cancer. We already mentioned the success of vaccines against cancer-causing viruses. Now we have things like cellular T cells. This is amazing science. You take a patient's own T cells, genetically engineer them in the lab to express a special receptor, a chimeric antigen receptor, or pull a tire that specifically recognizes an antigen on the surface of their cancer cells. Then you grow billions of these engineered cells and infuse them back into the patient. It's like giving their immune cells a new GPS system and weapons specifically targeted at the cancer. Another huge breakthrough is immune checkpoint inhibitors. These are antibodies that block the off switches, checkpoints, that cancer cells often use to suppress T cell activity. By blocking these breaks, the T cells can stay active and attack the tumor. And finally, there's targeted disruption of cancer. These are drugs designed to specifically interfere with the molecules or pathways that drive those cancer hallmarks. We discussed the oncogenes, the growth factor receptors, the angiogenic signals. Think of imatinib targeting the BCR-ABL fusion protein in CML, or antibodies against HER2 in breast cancer. These targeted therapies often have fewer side effects than traditional chemo because they're more specific to the cancer cells. The future is likely about combining these targeted drugs, maybe even with immunotherapy, to attack multiple hallmarks simultaneously, truly delivering on the promise of personalized medicine. So if we sort of connect all this back to the bigger picture, what we've really explored today shows cancer not as a single entity, but as this incredibly adaptive, diverse collection of diseases. Each one is constantly evolving its strategies for survival, growth, and spread, driven by those underlying hallmarks. We've journeyed from the basic definitions and classifications, right through the intricate biology of the oncogenes, the tumor suppressors, the immortality, the angiogenesis, the metabolic shifts, the immune evasion, the metastasis, then looked at how these manifest clinically in patients, and finally, how we approach diagnosis and the ever-expanding toolkit of treatments, from surgery to CRT cells.
Yeah, exactly. So what does this all mean for you listening? I think the understanding that cancer isn't just one disease, but many different diseases, each with potentially different combinations of these hallmarks driving them, that understanding is really the game changer. It's what's fueling the whole shift towards personalized medicine, where we look at the specific molecular profile of your tumor to guide therapy, making treatment potentially much more precise and hopefully more effective with fewer side effects. And just think about how far we've come. From Hippocrates seeing a crab centuries ago to now literally engineering a patient's own immune cells to hunt down cancer or designing drugs to hit incredibly specific molecular targets. It's astounding. Yet, challenges remain, right? Things like cellular dormancy, the incredible plasticity of the tumor microenvironment, the constant evolution of resistance. They raise profound questions. What does a cure truly mean in the face of potential dormancy? And how will our deepening understanding continue to reshape what's even possible in fighting these diseases? Well, that's our deep dive for today. Thank you so much for joining us as we explored the fascinating and sometimes daunting world of cancer biology based on understanding pathophysiology. We really hope you feel a little more well-informed and perhaps a lot more curious.